Now, in this part of our notes, we're going to do a Punnett square just review, and we're going to talk about a test cross. And eventually we're going to talk about how you can use chi-square to determine whether you're guessing right about the genetics of a trait or you're guessing wrong. To start on this page, we're going to go to the middle to practice 4 here and do a Punnett square related to blood types. Remember, with the blood types, we have A and B and O. A and B are codominant, and O is recessive to both A and B. If it says that we have a cross between a person that's heterozygous for A blood, what that means is that they have a code for A, and in the textbook you're going to see them write the code for A like that, stands for immunoglobulin A, and then they have a code for O. Again, if you're doing these problems and you want to write A and O, that's fine. It's up to you how you want to write your letters. Just make sure you know what those letters mean. A person that's heterozygous and they have B blood is going to have a code for B, and then also a code for O. If they had A, they would be, if they had B and they had A, they would have AB blood. So we know that they have a B and the other one has to be an O and not an A. Now, a Punnett square just helps us figure out what can get passed on. So sometimes this individual will pass on their code to have B blood, and sometimes they'll pass on a code for O. This individual sometimes can pass on a code for A, and sometimes it packages up a code for O. Bring them down, take them across. So we have AB blood. This is going to be an A blood type. This is going to be a B blood type. And that's going to be an O blood type. I'm going to change this a little bit if it hasn't already been changed for you. I'm going to change this to ratio so we can practice doing a ratio again to review. So when it says genotypes, we're going to be writing the letters. Well, we get 4 different combinations, and it says as percent. So I'm going to write 25%. I'm going to write the genotypes, A and B. We have 25% with A blood. So we have, again, we're writing the genotypes. 25%, that is B, that code for O. And then we have 25%, that would be. Okay, now it's going to ask for, in this case, phenotype ratio. In order to do the phenotypes and the ratio, you're going to have to start with the fractions. We have 4 different blood types. 1 out of 4 is big A, little a. 1 out of 4 is A blood. 1 out of 4 is B blood, and then 1 out of 4 is the O blood. So what you do is you make sure your denominators are the same, which they are, so then you just would take the top number. So it would be 1 AB blood to 1 A blood to 1 B blood to 1 O blood. Now, the next thing we're going to look at is when we're doing a test cross, what does that mean? Sometimes we have an individual and we're not sure of their genotype. If you remember, we had tall and short plants, and tall was dominant to short when it came to pea plants. Well, if we have a tall plant here, just by looking at it, we're not sure what it is. It could be big T, or it could be big T, little t. Okay, so that's what we call this the unknown. So here's your unknown plant. We know they're tall, but we don't know if they're homozygous or heterozygous. Okay, what we can do is we can do a test cross. So we're going to cross this with another individual, and we are always going to use a recessive individual. So in a test cross, what that means is happening is you have an unknown. That would be like our tall plant. And we will always cross the unknown with the homozygous recessive individual. In this case, it's going to be the short plant, or the little t, recessive looking plant. Now, if we make this cross, and again, we're crossing a short plant, and then we're crossing our tall plant. But remember, the tall, we're not quite sure what the tall one was. If you end up looking at the offspring, and all the offspring, 100% of them come out tall, then that means that this tall one was homozygous. They were big T, big T. Because if they're big T, little t, and they had several offspring, they would probably then end up with a short one, but they never did. If, again, we have taken that tall plant and we've crossed it, and we end up with about half the offspring are tall and about half the offspring are short, then that tells you that the tall plant had to be heterozygous. So again, what we're trying to do is figure out an unknown, and researchers are always going to cross an unknown with your homozygous recessive individual to help you determine that unknown. If all the offspring end up tall, then we know that the unknown was big T, big T. If about half and half, then we know they're heterozygous for that trait.